Actually, on a vacation during grad school, I dedicated about a week of my life to mining all of the palindromes out of the Google Voice database. So I actually obtained 3 of them. Um, well, thanks for the introduction, and I'm going to be talking today about a technique and then an application of the technique. Um, and first, disclosure. Well, we got a little Windows glitch there, but the as Manoj mentioned, I'm also the CEO of Microbiome. Um, and also, I just wanted to mention climate disclosure, that I offset the price, the carbon emissions from my flight for this conference. Costs about $5 on terrapass.com. And Microbiome is committed to offsetting all the carbon emissions for all the flights. So check out terrapass.com. It's kind of cool. So I'm going to talk to you a little bit about what I call the microbiome information gap. And this is something that I encountered when I started off in the field. I was a PhD student, and I was really excited to apply some techniques from machine learning, and I started to realize that the data we had were kind of crappy. We either had low resolution 16S data that gave us genus level profiles, or we had very high resolution deep shotgun sequencing data, but those tended to be so expensive that people wouldn't have enough samples in the study to really power studies. And so one of the things that I worked on once I started my own lab at the University of Minnesota was trying to fill in some methods here. And kind of thinking about it from a big data perspective. And what we realized is that you can actually get way more information using shallow shotgun sequencing than you would get for about the same price spent on 16S marker gene sequencing. And this was kind of revolutionary for us in our own studies, because it allowed us to kind of stop doing this two-stage system where we would do, the 16S first on the whole study and then zoom in with shotgun. Now we just do mid-depth or shallow shotgun sequencing on the whole study. And we did some validation of this. Here's the original paper on it, but we can see that you could have very deep data or absurdly shallow data of 500,000 reads, for example, and you capture the exact same signals in beta diversity, differences between communities. Same thing with alpha diversity. Even with as few as 10,000 reads per sample, you can pretty much nail the alpha diversity. These are species profiles of mock communities using deep and shallow sequencing. We can also recover species level biomarkers using shallow sequencing. This is a comparison of the p-values on the significant species between normal and type 2 diabetes subjects using deep and shallow shotgun sequencing. So this was really exciting. Oh yeah, we also sequenced the deepest sequenced human stool shotgun data ever. I was talking to Rob Knight, who was my PhD advisor, and he said, "Hey, I've got like $100,000 of Illumina reagents sitting on the shelf that are about to expire. What should I do?" And I said, "Why don't you just put a human stool sample on an entire HiSeq run?" And he did. So we ended up getting 2.5 billion reads per sample for a couple of samples. And this is a plot of the species abundance of different species in that sample using the shallow shotgun data versus using the entire 2.5 billion reads. So there's the shallow shotgun and the deep data. On the left is the species, on the right is KEGG orthology group genes. So we're actually getting quite good profiles for the known genes and the known strains and species. And what we're doing is taking advantage of miniaturization and automation to make the wet lab steps cheaper. And then we're usually getting somewhere between 2 and 5 million reads per sample, depending on the sample type. We also use a computational approach that extracts maximum information from the data. We have an alignment algorithm that's fully exhaustive, fully optimal, and it uses an entire read from the data to compare to every possible strain in the database. Always finds the best match. That's open source. If anybody wants to try it out, it's kind of fun. The benefit of this is that it fills in this information gap. For known species and genes, you're getting almost the same information as with deep shotgun sequencing. Of course, what you can't do with it is you can't assemble data and find novel genes, novel strains, novel species, and things like that. And just to be clear, more data is always better. I would never advocate for less data. And if you can afford deep sequencing on a very large study, you should do it. But the point is that it's usually more important to have more subjects and more samples than it is to go deeper on a small number of samples. So when you're allocating funds or planning to fund a study, it's usually more important to increase samples and subjects. So I'm going to talk now about an application of this in a study that recently came out of my academic lab. This is work that was led by Abby Johnson. She has a PhD in nutrition, but also knows how to code. And so she's been working in my lab on integrating diet data. Diet is the main driver of human gut microbiome variation. Do you agree or disagree? This is actually not entirely known or settled in the field, and you can find plenty of evidence for each of these conclusions, showing that diet dominates genotype, or finding that a very severe dietary intervention basically leaves people still in their own microbiome cloud. So we wanted to kind of get to the bottom of this with using this new technology we had. So as soon as Abby joined the lab, we collected a ton of data, and I'm showing you part of those data here. This is a representation of shotgun sequencing at every day for 17 days from 34 people. And you can see these are the species level profiles in each person varying over time. And it was the first time that I had seen species level variation within a person at this type of granularity. And we did this with shallow shotgun sequencing, so we were able to do this for about the same cost as a single deep shotgun sample, conventional deep shotgun sample per person. And there's a lot of interesting things we can do because we had that longitudinal data. Of course, I said we were interested in diet, so we collected a complete dietary record from every day from every person, and these are those data paired up. And there are a few things you might notice about the diet. If you ignore these two pink bars here and look at the other ones, what would you conclude about the variation of diet as compared to variation in microbiome? It's actually more variable. And this is like phylum level in diet. I mean, the orange color is just grain product. So then we realized we had a very interesting sort of, it's like multi-omics, but it's food omics, combined with microbiome problem of integrating these two data sets. Now, I mentioned these two abnormal subjects here. Do you guys know what happened to them? They were computer scientists, which apparently Abby told me this is what happens when you have computer scientists in your study. They consumed only Soylent for the entire, and I can say that I'm a computer scientist too. So they presented sort of an interesting sub-study because their stability was no greater or less than the other subjects, and they didn't converge. They maintained their individuality. So with this dense longitudinal data, we can do all sorts of new things. We can measure the stability within each person. And we found that stability, so this is your own microbiome variation from day to day, or lack thereof, is very strongly correlated with the diversity of foods you tend to eat on a given day. That was something new. And then we tried to answer these questions about does diet variation drive microbiome variation. And we can actually separate that into the two components. So understanding variation between people. These are the microbiome clouds of each person. So you can see each person has their own kind of individualized cluster. Some people are more variable, some people are less variable. And then on the right, I'm showing the personal diet clouds. And we wanted to try to integrate these data. But we realized we had a very tricky problem on our hands because the diet data was so variable. And this is Abby's diet. She had the most stable microbiome. She was very proud of that. And that's her diet at this very high level. And it's so much more variable than the microbiome. Here it is at the actual food level. And now it just looks like Picasso. And it's not clear how we're going to deal with all of that variation. So we looked at only the diet data. And this is where, for me, this is the type of project I like to work on because it's big data and we can make cool visualizations. So this is the first time I'd seen kind of natural diet data, what do people tend to eat? There was no intervention here, viewed in this way. The white points in the center are the subjects, and then the little colored dots are the foods they're eating, and then the foods are trying to be close to people who eat them and far away from people who don't. Can you find the Soylent drinkers, the computer scientists right down here? It turns out that's those two in the lower right-hand corner. One of them had pizza one time, and one of them had a Red Bull one time, and so it messed up the plot. They should have been off on their own. But we realized the diet data was actually highly personalized. There are some foods in the middle that many people ate, but most of the foods were out on the edge where only a few people ate them. How do we deal with this highly personalized nature? Well, conventionally, the approach has been to project those foods down into nutrient space, which would be great if we knew everything that was in the foods. But of course, we're only tracking the 100 or so nutrients that we know the human body cares about. We're not tracking everything that's in the foods that the microbes care about. So we tried to pair the microbiome with the nutrients. We took everyone's average microbiome throughout the study, made the cloud, took their average nutrient intake throughout the study, made that cloud, and then we used this analysis called Procrustes, where you kind of rotate one to match the other. And we found that it was kind of a hairball. They didn't really correspond. And then we thought, okay, this actually kind of makes sense. We need to use the foods instead of the nutrients. So Abby developed this method called Food Tree that kind of organizes similar foods together and lets us use tools from microbiome analysis that leverage phylogenetic trees. So now we pair microbiome with foods, and actually it's a strong correspondence. So the food intake explained about 19.4% of variation between people. We also looked at variation within a person. So does your daily choice of food impact your microbiome? And we did the same analysis with the daily variation. So now we're showing the same Procrustes analysis, the first coordinate of that against time. And we found that the vast majority of subjects actually had a significant correspondence with the daily intake of foods over time. And that explained about 24% of the variation. But because we had this dense longitudinal data, we could see that in one person, a certain type of food would cause a certain species to grow, and another, and such as this cakes, cookies, bars, was associated with growth of Roseburia inulinivorans, which consumes inulin, a common food additive. And in another person, sometimes the signal would go the other way. So there were some generalized signals and some personalized signals. So we found that microbiome diet associations were personalized. And this is something that we couldn't really have understood without that dense longitudinal data. And there are a few different causes that we're working on now. So in conclusion, the main point that I'm trying to leave you with is that scalable shotgun sequencing can enable new types of studies with very high resolution and dense longitudinal data. And then I showed you an example where we saw a diet actually explains about 20% of the variation between, 25% of the variation within people, and that the foods, not the conventional nutrients, explain most of the microbiome variation. So we also need more work in the diet space, which is a topic for another day. Thank you very much.